Okay, let's dive in. Cancer. It's just so frustratingly good at dodging our body's defenses, isn't it?
It really is. And, we've made these huge leaps with immunotherapies, getting our own immune system to fight back.
Right, therapies that have been game changers for many,
Hmm.
but they don't work for everyone.
No, exactly.
Oh,
Especially with some of these really stubborn, tough-to-treat cancers, there's still this big question mark. Why do they fail sometimes?
why indeed? And that's where our source material today gets really interesting. It seems to point towards a kind of, well, a hidden mechanism cancer uses. Something involving fat metabolism inside the immune cells themselves. Seems unexpected.
It does, but it makes a lot of sense when you dig into it. What the research suggests is that immune cells inside the tumor start taking up fat.
Okay.
And this isn't just, fuel. It acts like a metabolic checkpoint. It essentially paralyzes the body's anti-cancer response right there in the tumor.
A metabolic checkpoint.
Mm-hmm.
So not like the usual checkpoints we talk about, the PD-1, CTLA-4 type things.
Exactly. Think of it less like a protein signal on the surface and more about the cell's internal. Well, its fuel system, its metabolic state being manipulated.
Huh.
And the sources really zoom in on one particular protein here, CD36. It's a key transporter for fat.
CD36. So that's the gatekeeper letting the fat in. And that fat somehow gums up the works for the immune cells.
That seems to be the core idea. And crucially, the sources also highlight a potential way around this specific problem.
Ah, okay. A potential solution.
Yes, a new antibody they've developed. It's called PLT012, and it's designed to go right after that CD36 protein on the immune cells.
Right. So our mission for this deep dive, then, is to unpack how this metabolic trickery actually works, what this PLT012 antibody does exactly, and what the early results are telling us. Sounds like some promising stuff in the source material.
Definitely worth digging into. Let's start with how the cancer hijacks things metabolically. The sources paint this picture of the tumor microenvironment.
Right. The neighborhood the tumor sets up for itself.
Exactly. And it's often acidic. And here's the key bit. It has high concentrations of specific types of fats, lipids.
Like a toxic swamp, but specifically tailored with fat.
That's a good way to put it, yeah. And in that specific fatty environment, the sources show that different immune cells actually start expressing more CD36 on their surface.
So they put up more gates to let the fat in? They're actively importing it?
Seems like it. CD36 fundamentally brings certain fats and cholesterol into cells. But here's the really surprising part the sources highlight.
Okay.
This flood of fat, it has vastly different effects depending on which immune cell we're talking about. And for the cells we want fighting the cancer, it's bad news.
Different effects. So the fat isn't just bad across the board.
Not at all. Get this. The sources show that cells like regulatory T cells, TREGs, and these myeloid-derived suppressor cells, or MDSCs.
Those are the ones that naturally dampen immune responses, right?
Okay.
Brakes.
Exactly, the brakes. Well, they actually thrive on these lipids. The fat metabolism seems to boost their suppressive function. It makes them better at shutting down the anti-cancer attack.
Wow. So the tumor's fatty environment is literally feeding the enemy within, making the suppressors stronger.
Precisely. And it gets even more intricate. Macrophages, those really adaptable immune cells.
Yeah, they can be good guys or bad guys depending on the signal.
Well, this fat uptake seems to reprogram them. Instead of potentially being anti-tumor, they shift towards a pro-tumor state. They start helping the cancer grow and survive.
Okay, so it's feeding the suppressors, and it's turning potential fighters into collaborators. This fat is really doing a number on the immune response.
It really is. And then there are the main soldiers, the CD8 plus T cells. These are the primary killers we rely on.
What happens to them?
The sources indicate they suffer profound dysfunction. They essentially get overloaded by this fat uptake via CD36, and it drives them towards a specific type of programmed cell death.
Programmed death.
Yeah.
You mentioned ferroptosis earlier. Is that it?
Yes, exactly. Ferroptosis. It's linked to iron and lipid breakdown products. So these critical killer T cells, by taking in too much fat through CD36 in that environment, are basically pushed into self-destruct mode.
So the main fighting force is being taken out by its fuel source, essentially.
Yeah.
The fat it's forced to take in.
It's a really effective way for the tumor to create this immunosuppressive shield. It's not just blocking signals, it's manipulating the very metabolism of the immune cells.
This CD36 seems like a really critical linchpin in this whole process, then.
It certainly appears to be a key player in establishing this metabolic barrier. which naturally brings us back to that potential solution mentioned in the sources, the PLT012 antibody.
Right, the one developed by the teams at Ludwig Cancer Research and Taipei Medical University. You mentioned Ping Ji Ho and Yiru's labs.
That's the one. And its specific target is CD36. But importantly, the sources stress it targets CD36 on the immune cells within that tumor microenvironment.
Ah, okay, so it's not hitting CD36 everywhere, but zeroing in on the immune cells caught in that fatty trap.
That seems to be the design goal. And the mechanism, as described in the sources, is pretty straightforward. It blocks CD36.
Just blocks it, stops it from working.
Yeah, it physically prevents CD36 from grabbing onto and importing those fatty molecules into the immune cells. It shuts down that specific gateway.
Okay, so if the fat can't get in, does that actually reverse all those negative effects you described on the tregs, the macrophages, the CD8s?
Well, that's what the preclinical data strongly suggests. By blocking CD36 with PLT012, the sources show it selectively cuts down that lipid overload in the tregs and MDSCs.
So they can't use the fat to power up their suppression anymore.
Seems like it dampens that effect, yes. And critically, for the CD8 plus T cells...
So killer cells.
Yeah, the sources highlight that blocking CD36 appears to preserve their function. It stops that dysfunction and that ferroptotic cell death pathway. It lets them survive and, hopefully, regain their ability to attack the tumor.
That is fascinating. So it's less about adding more soldiers and more about changing the battlefield conditions, the metabolic environment, so the soldiers already there can actually fight.
Exactly. You could say it acts upstream. It's modulating the lipid metabolism itself. The sources contrast this with, say, conventional checkpoint inhibitors.
Right, the ones that take the brakes off T cells.
Yeah, but those might struggle if the T cells are already metabolically crippled by this fat overload before you even take the brakes off. If the engine's seized, releasing the brake doesn't help much.
So this PLT012 approach tackles the seized engine problem first by fixing the fuel issue.
It aims to dismantle that specific metabolic checkpoint, yes. And the preclinical evidence reported in the sources looks quite encouraging on that front.
So what did they find in the lab? You mentioned mouse models.
They did. They used models of liver cancer, hepatocellular carcinoma, or HCC, and also colon cancer that is spread to the liver. In those models, PLT011 reportedly restored anti-tumor immunity.
Not just restored the immune cells, but actually impacted the tumors.
Yes, the sources state it significantly inhibited tumor growth in those models. And another key finding was synergy.
Synergy, meaning it worked well with other treatments.
Exactly. It showed strong synergy when combined with existing checkpoint inhibitors and with other standard cancer therapies. They worked better together than alone.
That's always a huge plus, right? Anything
Sure.
that can potentially boost current treatments is important. What about, safety and relevance for actual people?
Good question. The sources do address that. First, they apparently took tumors isolated from human HCC patients and showed in the lab that the antibody could reshape the immune cell landscape in those human samples, too.
Okay, so it worked on human cells outside the body, which suggests it might translate.
It points in that direction, yeah. And then there were preclinical safety studies in monkeys and mice. These reportedly showed a favorable safety profile. Now, this is important because CD36 isn't just on immune cells in tumors. It's expressed quite widely in the body.
Right, so targeting it. You'd worry about hitting healthy tissues, causing autoimmune problems.
That's always the concern with broadly expressed targets. But the sources mention the antibody's rational design.
Meaning they engineered it carefully?
Apparently so, engineered in a way that seems to allow it to be tolerated, which is obviously crucial if it's going to be used as an immunotherapy in people.
Okay, so we've got this problem. Cancer using fat and CD36 to shut down immunity. We've got this potential solution, the PLT012 antibody blocking that process. It looks promising in preclinical tests, seems reasonably safe in early studies. What's the bigger picture here? What does this mean for cancer research overall?
Well, I think what's really significant is that these findings really establish CD36 as a targetable metabolic immune checkpoint. It's not just about protein interactions anymore. We're looking at the metabolism itself as a checkpoint.
A whole new category of target, potentially.
Could be. And the sources position PLT021 as possibly the first in a new class of immunotherapies, ones that work by tweaking immune cell metabolism.
And is this moving forward
Sure.
towards human trials?
It seems so. The sources mention it received an FDA orphan drug designation that helps support development for rarer conditions like potentially certain liver cancers, and that a spin-off company is taking it forward for clinical evaluation.
That's a big step. Getting it into human studies is key.
Absolutely. And the potential implications might go beyond just liver cancer. While the initial studies focus there, the sources note that CD36 is found on immune cells and lots of other tumor types too.
Especially ones known to have these lipid-rich microenvironments.
Exactly. So this strategy, blocking CD36-mediated fat uptake, it might be relevant for a whole range of other cancers that are tough to treat with current immunotherapies.
So it's not just a potential new drug, but it also gives us this deeper understanding of how tumors fight back using metabolism.
Definitely. It highlights that tumors are sophisticated metabolic engineers, actively disarming immune cells by messing with their fuel supply.
Okay, so wrapping up this deep dive. The core idea, the big takeaway, is that going after the metabolic function of immune cells, specifically targeting CD36 and fat uptake, could be a really novel and hopefully powerful way to unleash the immune system against cancer.
Yeah, particularly for those cancers that have proven resistant to our current immunotherapy strategies. By disrupting this metabolic blockade, you might just restore the immune system's fighting chance.
And for you listening, understanding these really intricate interactions, this crosstalk between tumor metabolism and immune function, it's pretty crucial for appreciating where the cutting edge of cancer research is heading and what future treatments might eventually look like.
It really shows how the field is broadening its focus, isn't it?
Yeah.
Looking beyond just the cell surface signals and digging into the metabolic nuts and bolts within the tumor environment itself.
Which definitely leaves us with something to think about. If cancer is this good at hijacking one metabolic pathway, like fat uptake via CD36, well, how many other metabolic tricks, other metabolic checkpoints might it be using? And what other amazing strategies might we develop by targeting those in the future?